Coming up next, we have two very special guests that are here to talk about a topic all of us don't really know about. Stay tuned for more. Don't go anywhere.
Hi, I'm here with Dr. Cristina Uribe.
And I'm here with Dr. Paula Jaramillo, and we're here to tell you about the bone marrow stem cell transplant.
To start us off tonight, the bone marrow is a spongy tissue found inside the bone. It contains cells known as hematopoietic or the blood-forming stem cells. These cells divide and mature into three types. The first type is known as the white blood cells, which are in charge to fight infections. The two types of white blood cells are called neutrophils and lymphocytes. The second type is known as the red blood cells, and it is used to carry the oxygen throughout the organism. And the last type is called the platelets, and it is used for blood clots.
Stem cells are unique and unspecialized self-renewing cells with the potential to develop into other types of cells. The bone marrow is the primary place where stem cells are located and produced.
Diseases the stem cell transplants are used for are primarily blood disorders.
Depending on who the donor is, there's different types of transplants.
There are three types of transplants. Autologous, in which the person receives their own stem cells. Syngeneic, where patients receive the stem cells from their identical twin. And allogenic, where the person who is related like a brother or sister donates to the patient. It can also be an unrelated donor or even an umbilical cord stem cell.
We all have different sets of protein. The set of proteins called the HLA type is identified by a special blood test. The higher the number of HLA antigens, the greater the chances the patient's body will accept the stem cells.
The patient receives the stem cells through an intravenous line. Proteins bind to receptors of the frizzled and the LRP families on the cell surface.
The signal is transduced to a beta-catenin. It is transduced, it enters the nucleus and forms a complex with the TCF to activate transcription of HLA target genes. HLA target genes can also signal through the tyrosine kinase receptors.
During the growth of the tissues, these ligands and receptors are transcriptionally controlled by the signals to ensure balance between proliferation and differentiation. They act to maintain the undifferentiated states of stem cells.
Other growth factors instruct the cells to proliferate or multiply. These other factors include FGF and EGF signaling through the tyrosine kinase pathways.
There are two types of the cells in the bone marrow. These are called the stromal and the osteoblast, which produce a protein called SDF1 that attracts them. Endothelial cells in the blood vessels capture the SDF1 and release them into the bone marrow.
Because of these, the protein is then released, a chemotactic gradient is formed with high concentrations of SDF1 and is located very closely to the stromal and osteoblast cells.
Then, the SDF1 interacts with the CXCR4 receptor that is located on the surface of the stem cells, bone marrow endothelial cells, and other stromal cells.
Because of these, the SDF1 creates a bond with the CXCR4 that signals the stem cells inside the bone marrow to move towards the stroma and osteoblast cells. The stem cells cannot leave the bone marrow through the blood.
Normally, a small amount of stem cells leave the bone marrow and travel through the body in its circulating blood, where they assist in repair and defense of the body.
After chemotherapy or radiotherapy, transplanted blood stem cells are put into the bloodstream that starts producing new, healthy cells. Capillaries are 1 cell thick and made of endothelium. This makes them leaky, meaning that small molecules like water or ions can diffuse through. Sinusoidal capillaries found in the liver, bone marrow, and other places are a special type of fenestrated capillaries but have larger openings in the endothelium that allow red blood cells, white blood cells, and serum proteins to pass through. The size of an opening is approximately the size of a red blood cell.
Stem cells have been through both plant and animal kingdoms. During the development of multicellular organisms through natural selection, the stem cells went with the process.
There is a probability in which a stem cell gives rise through asymmetric cell division, splitting into a stem cell daughter, while the other undergoes differentiation. Another route of cell division that stem cells follow is stochastic differentiation. Stem cells make a combination of asymmetric divisions and symmetric ones. they produce are two stem cells or two differentiated stem cells.
Research really never stops, and we're always trying to find new ways to help.
Thank you for letting us be here today. I'm Dr. Cristina Uribe.
And I'm Dr. Paula Jaramillo.